Hello students, in previous part we have discussed about the inducible operon or lag operon. In this part we will discuss on suppressible operon. Suppressible, or we can say that repressible. Repressible operon. This is also known as tryptophan operon. Tryptophan operon. Tryptophan is also an enzyme which is responsible for the growth of the cell. How this tryptophan is regulated? In normal condition, we have seen that this operon remains on. Normally, it remains on mode. How this on mode remains? Suppose a cell is produced tryptophan and this tryptophan is utilized by the cell in growth. Then the process remains continued. How this process can remain continued? We have taken the process of a piece of DNA which contains two parts. One contains regulatory gene and other part contains structural gene. The regulatory gene which we have discussed just like in lag operon, promoter, but in place of inducible, in this the regulatory gene is a repressible gene R in place of inducible I. Again promoter and then operator. If a cell tryptophan synthesizes, how the tryptophan synthesis, the process which we know that the cell is also required to grow, and to grow cell take the energetic particle lag operon into glucose and galactose and tryptophan. This is the part of regulatory gene and there are 5 structural genes, and those structural genes are TRPA, it means tryptophan A, tryptophan B, tryptophan C, tryptophan D and tryptophan E. These 5 are structural gene and these are regulatory gene. RNA polymerase first of all attaches with the promoter gene. RNA polymerase. One RNA polymerase attached with the promoter, this one, and another one here. As the promoter gene gain RNA polymerase, the process starts. It means this gene slide the RNA polymerase from this one to another. Next slide. Here R. What is the role of R? The role of R just like as inducible gene, and that means the DNA converts into mRNA and mRNA converts into protein. The process in which DNA converts into mRNA, that is transcription, and mRNA to protein is known as translation. During a normal condition, if the tryptophan is synthesized, then the protein which is synthesized by the repressible gene, suppose this is the protein, this protein remains inactive. A major difference between lag operon and tryptophan operon, the protein which is synthesized by the inducible in lag operon, that is activated. But if the protein which is synthesized by the repressible, that is inactive, and this inactive protein is called a apoenzyme. This is inactive protein, and this inactive protein is called apoenzyme. If this protein cannot be bind with operator gene, cannot be bind, it means the process remains continuous. So it remains in on mode. Always remains on mode because the protein which is synthesized by the repressible gene is inactivated. Due to inactiveness, it doesn't bind with the operator gene. and this is called apoenzyme, because we know that all enzymes are made of protein. This is the protein. Then what are the role of structural gene? There are 5 structural genes, and these 5 structural genes synthesize different types of enzyme. But collectively, the enzyme which is synthesized by all 5 structural genes, that is called tryptophan synthetase. This will also synthesize an enzyme. These all different tryptophan parts, structural gene contain different synthesize different enzyme. But collectively, the enzyme which is synthesized by the whole structural gene, that is tryptophan synthetase. Tryptophan synthetase. This activate or this synthesize the tryptophan. Tryptophan. Again listen. As the process remains continued, these 5 structural genes synthesize collectively tryptophan synthetase enzyme. This tryptophan synthetase enzyme produces tryptophan. This tryptophan is used by the cell for growth. So this process remains continued. It is called on-mode. As we know that if the process remains continued, one stage will become the amount of tryptophan enzyme is exceed. It means it forms extra gene, and these extra gene combine with the protein, this one, and make it activate, which bind with the operator gene, that they change into off mode. In on mode, this structure remains same. But as we discussed on off mode, how this process occurred in off mode. We know that repressible or suppressible operand in normally condition remains on. We turn to off mode. How this off mode can be taken place? Again, there are the structure of same, the structural and regulatory gene. The regulatory gene, which is promoter by repressible, promoter and operator. RNA polymerase 1 attached with the promoter here, and the another one is here. This is the RNA polymerase. And the 5 structural gene remains such: this one, tryptophan A, tryptophan B, tryptophan C, and tryptophan D or E. These are the 5 structural genes. As the promoter gain RNA polymerase enzyme, the process of translation to translation will synthesize the protein. See, here DNA converts into mRNA, and this mRNA to protein. This form the protein. If this continues, the process remains same. It means the synthesis of tryptophan is just required by the cell amount remains same. Suppose there are 100 molecules of tryptophan required by the cell in normal condition. The cell requires 100 molecules of tryptophan, 100 mole of tryptophan, and the producer is also 100. The process remains on. But due to continuous synthesis, if one particle of tryptophan is also exceeded, it means one extra tryptophan is also produced. This extra rather than 100, because this 100 is required by the cell, this one extra tryptophan bind with this inactive protein. Firstly, it remains inactive. It is inactive. And inactive protein is known as apoenzyme. But as the one extra parts of tryptophan is produced, this extra one tryptophan bind with the protein, means apoenzyme, and it changes into activated protein. It changes to activated protein, and if this protein is activated, this protein binds with the operator gene, and if the activated protein binds with the operator gene, the process of synthesis of tryptophan stops, because there is no production can take place in this structural gene. स्ट्रक्चर जीन में किसी प्रकार का प्रोडक्शन नहीं होगा तो क्या होगा, यहाँ पर किसी प्रकार का सिंथेसाइज ऑफ प्रोटीन और एंजाइम डजंट टेक प्लेस बिकॉज द ऑपरेटर जीन इज ब्लॉक्ड, एंड इन दिस कंडीशन वी नो दैट द प्रोसेस विल बी स्टॉप्ड, एंड दैट इज कॉल्ड ऑफ मोड. As the remains amount of tryptophan, extra tryptophan is utilized by the cell. Again, suppose it contains 101, required by the cell 100 particle. It means one particle of tryptophan is extra. This extra tryptophan binds with the protein and activates it. This activated protein binds with the operator gene, and the process remains off. Suppose this 1 extra, 2 extra, 4 extra are utilized by the cell. Then it remains again 100. The process remains same because the 100 particles are required, and the all extra tryptophan is deleted or utilized by the cell. Only remains 100. We know that this is the imaginary condition, not all time required 100. The imaginary, how the tryptophan particles are required by the cell. It depends on the nature and how the growth can be take place. Extra tryptophan bind with the protein, activate it, and this activated protein bind with the operator, process stop. If the extra tryptophan deleted, then the remains required tryptophans, again this process becomes on. So this is the operator, this is the regulation in the tryptophan operon. We have seen that there are two operon. One is inducible normally remains off, it turns to be on, and second one is repressible, suppressible and tryptophan operon. In normal condition it remains on and it turns to be off. In next part we will discuss on HGP, Human Genome Project.